Okay, so we're going to start chapter 10 today, the citrate cycle. So we covered glycolysis, and we know at the end of glycolysis, we have pyruvate. And we did get a little bit of ATP in converting glucose to pyruvate, but the most ATP that comes from metabolism of glucose comes from the citrate cycle and oxidative phosphorylation. So you get a little bit out of glycolysis, but glycolysis is really great because you can do it anaerobically. So we don't call the citrate cycle an extension of glycolysis because it really initiates production of energy from multiple things, from fatty acids, from proteins, and from glucose. So really it's the link between many different metabolic pathways, and we use oxidation of whatever the fuel source is to produce ATP, right? And we know we're going to use NAD+, we're going to use FAD as electron carriers in this pathway. All right. So we're going to talk in general about how we're going to capture that energy, and then we're going to get into how we go from pyruvate to acetyl-CoA, because remember, that is not in the citrate cycle. This is technically, this step right here is not in the citrate cycle. It is required to run the citrate cycle, but it is not a part of the citrate cycle. Then we'll talk in detail about the citrate cycle. So we already kind of said this, that the citrate cycle captures energy using redox reactions. We've talked lots about redox reactions. So this is the hub, right? Because it's pulling metabolic fuel from different sources. So it's pulling it from amino acid metabolism, from carb metabolism, from fatty acid metabolism. So whenever ATP stores are low, the citrate cycle can use metabolites from those three various sources in order to generate ATP, right? And you can actually go in reverse too. So take biochem too, and we can talk all about it. So we also call this cycle the Krebs cycle, or the tricarboxylic acid cycle, right? And I prefer to say the citrate cycle, and the reason that I prefer the citrate cycle is because it tells you about the very first reaction, the product of the very first reaction, which is really, I feel like, more helpful than Krebs, which is a person who discovered it, and the tricarboxylic acid tells you about the structure of the molecule, but it doesn't tell you the substrate. Okay, so remember from chapter 2, right? We can couple oxidation reduction reactions and biochemical pathways to get energy for ATP synthesis in the mitochondria. So all of the enzymes in the citrate cycle and the electron transport chain reside in either the mitochondrial matrix, or in the mitochondrial inner membrane. Okay, so if it's in the mitochondria, then we know it is aerobic. We have oxygen present, and our goal here is to oxidize acetyl-CoA. So we made pyruvate, we made pyruvate in the cytosol. We're going to actually transport it into the mitochondria using pyruvate translocase, so that we now have pyruvate inside of the mitochondria. Then we're going to talk about that one reaction where you have to convert pyruvate to acetyl-CoA. Then acetyl-CoA can enter the citrate cycle. So when we look at just the citrate cycle, so all by itself, just the citrate cycle, what are we going to get out of the citrate cycle? We're going to get 8 electrons. So each turn, and remember, each turn is really one acetyl, E-T-Y-L, CoA, acetyl-CoA, and that acetyl-CoA came from glucose. But how many acetyl-CoA molecules are we going to get from glucose? We're really going to get 2. So every molecule of glucose will take us through 2 turns of the citrate cycle. But here we're just talking about 1 turn because it's kind of easier to see 1 turn. So in 1 turn, we get a transfer of 8 electrons. We're going to generate 3 NADH molecules, 1 FADH2 molecule, and 1 GTP, right? Okay, so why do we call it the citrate cycle? Because citrate is the conjugate base of citric acid, which is the first product in the pathway, right? So that's kind of the nice thing. All right, so here we go. This is the citrate cycle. We call this the metabolic engine because it kind of can relate to one. So what do you put into the citrate cycle? Well, our fuel is acetyl-CoA. So that's what we're putting into the cycle. And what are we going to get out? Well, we're going to produce some kind of work, but we're also going to have exhaust because like a typical engine, you have things that are produced that you don't want. So the things that you don't want are carbon dioxide. But the three things that we make, we just said, were GTP, FADH2, and NADH. And those are our sources of power for redox energy. And so that one turn of the citrate cycle, if I produce one GTP, one FADH2, and 3 NADHs, I'm eventually going to get to make 10 ATP by going through the electron transport chain and oxidative phosphorylation. So how does that translate? Well, if I look at my one GTP is a perfect translation. One GTP will get me one ATP. If I look at FADH2, my FADH2 is going to enter the electron transport chain. And so that FADH2 is going to give up its electrons, and those electrons are going to go through a series of linked redox reactions in order to generate ATP. So from one FADH2, I'm going to get 1.5 ATP, and that is supposed to be FADH2. Then from my 3 NADH, for every 1 NADH, I'm going to get 2.5 ATP. So for 3 NADHs, I'm going to make 7.5 ATP. So if I add up my 1 from GTP, my 1.5 from FADH2, and my 7.5 from NADH, I'm going to get a total of 10 ATP. So that's going to give me my end result, right? I want to make ATP. That's great. But what are some things that are really important for making this reaction happen? Well, we know that this reaction is cyclic, right? Everybody draws it as this circle, right? So it is cyclic. What does that mean? What does a cyclic pathway mean? That our substrate of our very first reaction has to be a product of the last reaction. And we see that in oxaloacetate. So oxaloacetate is the product of the very last reaction, citrate, and a substrate of the first reaction. And then acetyl-CoA. Acetyl-CoA is another substrate of that very first reaction. And so however much acetyl-CoA comes into the citrate cycle is going to control how much we run the citrate cycle. So we have two enzymes that actually control the flow of acetyl-CoA and oxaloacetate into the cycle. And those two enzymes are not a part of the citrate cycle, but we're going to talk about them at the end of the chapter when we talk about regulation. So those two enzymes are pyruvate dehydrogenase and pyruvate carboxylase. And we'll talk a lot about pyruvate dehydrogenase kind of in the beginning, and then we'll talk more about pyruvate carboxylase later. So this is kind of a nice little reminder, right? If we're going to be doing oxidation reduction reactions, you have to remember your oil rig, right? So whatever substance is giving up electrons, that is the reductant. Whatever is going to accept is the oxidant. So if we look at that reductant and it has lost its electrons, therefore that's an oxidation reaction. And then our oxidant is whatever picks up the electrons. So if we go from not having them to picking them up, then that is the reduction reaction. So we would say that A in this scenario is oxidized and B is reduced. But the reductant and the oxidant, it kind of sounds opposite, but it's not. Okay, so we said that we're going to use a lot of NAD plus and FAD in our redox reactions. And so these are really, really common pieces of enzymes that are in lots of different oxidation reduction reactions. They come from vitamins. And so how do we define vitamins? Vitamins are things that our bodies don't produce, but that we need in order to be healthy. So how have we defined vitamins in the past? If you have a metabolic issue, right, you're exhibiting some kind of symptoms, and if we can fix it through nutrition, then that's a vitamin. So niacin, vitamin B3, and riboflavin B2 are the sources of NAD plus and FAD. And so, you know, evolutionarily over time, we have lost the ability to make a lot of these things because we eat things in our diet that have them, and so we don't need them anymore. And so it's metabolically a waste of energy to make them when we're just going to eat them. So that's why it's really important to have a balanced diet and take your vitamins if you're going to have some kind of special diet or that kind of stuff. Okay, so NAD plus, when it goes to NADH, the reduced form, you're going to have one reaction. And in that one reaction, what's actually going to happen is that you're going to get two hydrogens from whatever your reductant is. And those two hydrogens are going to be kind of in a different form. So you have a normal proton here, but then you also have so this is a hydrogen with a negative sign and then two dots. So what this tells us is that we have one proton. That's what makes it the hydrogen. And then we have two electrons. So we have a net charge of -1. And so that's why we represent this that way. And so our NAD plus is going to pick up two electrons that way in one reaction. And you can see the lone pair on nitrogen and that you've picked up a hydrogen on this carbon. FAD will pick up the electrons in a two-step mechanism. So it picks them up one at a time. And so when it picks up just one, we represent that as the semiquinone. So you just put a little dot at the top at the end after FADH. And then the fully reduced form is FADH2. All right, so this is just kind of a reminder. Standard reduction potential. So this you should have kind of talked about before, but it's all about the amount of energy, right, that is stored, and whether we can transfer electrons or not. And so when you look at these different half reactions for redox reactions, right, you're always kind of keeping track of where electrons are going. And these are only half reactions. So you have to remember that in the cell, you're not going to have a single half reaction. You're going to have two halves that are coupled, and then the electron is the intermediate in between that gets canceled out that drives the reaction. Okay, so basically what we've done is taken at standard conditions right here. Excuse me, y'all. At standard conditions, we've taken the hydrogen half reaction and we've set that standard reduction potential to 0. And then we're comparing all the other half reactions that we find in the body. And so we can identify something as a stronger oxidant or as a weaker oxidant, right? So if we have an oxidant that has a positive reduction potential, then what can we say about its affinity for an electron as compared to hydrogen, right? Because we're always going to compare it to hydrogen in the first place. So if you have a positive reduction potential, then what you're going to have is a really high affinity for electrons. If you have one with negative, you're going to have a lower affinity. So things at the top, right, those are going to be our really, really strong oxidants. So the strongest oxidant, what do we find here, is O2. And we know that O2 is our final electron acceptor when we're in the electron transport chain. And so that makes sense. You want the last one to be the strongest oxidant. Okay, so you can look at whether or not a reaction is going to happen, a transfer of electrons is going to happen. And all you have to do is look at the acceptor energy minus the donors. And so it's a lot like delta G, right? But it's opposite. If our standard reduction potential is greater than 1, it's positive. If it's less than 1, it's not favorable, right? And you can actually really can relate our standard reduction potential back to our free energy, right? So this is just kind of to keep an idea of what reaction is favorable and what is not when we talk about transferring electrons. All right, so oops.